Hello, and thank you all for joining us this afternoon. In this session, we're going to be sharing a webinar which was developed by the Sustainable Spotted Wing Drosophila Management Team, which is supported by the USDA Specialty Crops Research Initiative. This webinar was originally recorded in December, and I'm going to be available to answer questions that come up during the session. So please, while you're viewing the session, go ahead and type any questions you guys have into the chat box, into the Q&A box, and I will handle those as we go through. I will also reserve some of those questions to talk through with you all at the end. Before we get started, let me just quickly introduce myself. I'm Hannah Burek. I'm a professor of entomology at North Carolina State University. And for the last 5 years, I've been coordinating this national collaboration to deal with the management, biology, and ecology questions that spotted wing Drosophila has raised in all of the small fruit crops that it impacts. The webinar that you're going to see includes information from collaborators throughout the country who are associated with the project, and they'll introduce themselves one by one as they go through each of the slides. So with that, we can go ahead and get the recording playing, and I'll look forward to seeing your questions in the Q&A box. Welcome, and thank you for joining us for our webinar on spotted wing Drosophila, Lessons from the Last Decade for Management. We're going to go ahead and get started. And I want to highlight that this is a project output associated with the Sustainable Spotted Wing Drosophila Management Project supported by the USDA NIFA Specialty Crop Research Initiative. This webinar will last about an hour, followed by questions from the group. We'll be posting a recording of today's webinar at our project website, SWD Management, next week. We don't have pesticide credits available for this session if that is something you're interested in. However, if you live in a state where you can file requests on your own afterwards, you can contact me, Hannah Burek, at the email listed here for documentation of your attendance. Thank you so much for joining us. If you have questions during the webinar, you can type your questions into the Q&A, and the moderator will collate and ask these questions at the end of the session. After the webinar, you can direct any questions to myself, again, at the email listed on the slide here. The presenters for today's session include collaborators from our large Sustainable Spotted Wing Drosophila Management Project, myself, Hannah Burek, based at North Carolina State University, Greg Loeb from Cornell, Joanna Chu from UC Davis, Vaughn Walton from Oregon State University, Phil Fanning from the University of Maine, Rufus Isaacs at Michigan State University, Kent Dana from UC Berkeley, Ash Sayal from the University of Georgia, Cesar Rodriguez-Sona from Rutgers University, and Miguel Gomez, also at Cornell University. Our outline for today is ambitious, so our goal is to cover the progress that we collectively have made on understanding the biology and management of spotted wing Drosophila over the last roughly 10 years since its introduction and global range expansion. To do this, we're going to be talking about several subject areas. First, invasion biology, the seasonal biology of SWD, and how its population genetics might inform our management methods and tactics. Next, we'll discuss host range and preference, followed by pesticide efficacy and non-target effects of those pesticide management tools. Finally, biological control, cultural and post-harvest controls, monitoring and applications for behavioral control, and finally, putting the pieces together to help use all of this information to make better management decisions. As I said, this is an output of a large, nationally representative research collaboration that is led by Ash Sayal, based at University of Georgia, in collaboration between many of the presenters in this session, as well as others from around the United States. This project will be continuing for the next 4 years to further our understanding of spotted wing Drosophila biology and management. The goals of this project are broadly to assemble teams of growers to implement our best management practices for spotted wing Drosophila and to really serve as a test lab for those management practices, to develop decision aids for farmers to increase profitability, implement sustainable alternatives to insecticide usage, to reduce the risk of insecticide resistance, and then to disseminate this information to enable producers to optimize their pest management decisions. And that's what we're going to do today. You'll also find more information about our project at our project website, swdmanagement.org. So without further ado, I'm going to turn it over to Greg Loeb, who's going to cover our first topic, invasion biology, seasonal biology, and population genetics. And Greg, just let me know when you're ready for me to advance slides.
Good, thanks, Hannah. I'm gonna make this a pretty fast summary of some of the work we've been doing on trying to understand the factors that drive population dynamics of spotted wing and risk, and therefore hopefully lead to better pest management. The three sort of subtopics I'm going to talk about are the effects of low and high temperature tolerances on spotted wing survival and its implications for seasonal biology. And I want to talk about some work and the outcomes of population modeling that we've been doing as a team, and how it can be used to improve pest management decision making. I'll give you a specific example. And finally, I want to talk about some of the work that we've been doing on the population genetics and the variation in genetics in spotted wing populations and what that has to say about how spotted wing moves, and that has implications for pest management as well. Now, you can learn more about a lot of these topics with our previous last webinar associated with this team, and you can see the link there at the bottom of this first slide, and Hannah referred to it earlier. Next slide, please. The pattern of population dynamics kind of describes the seasonality and also determines when you as growers are at risk for spotted wing Drosophila infestation. Spotted wing Drosophila is a temperate species of vinegar fly. It's kind of constrained in the north by the cold temperatures, winter temperatures in the Arctic, and constrained from in the south from probably high temperatures and maybe other factors. Within the temperate zone of the United States, we also see variation in the seasonality of spotted wing. In the area where I live, in the north temperate area, we typically see a large peak at the end of the field season, and then during the winter, we virtually can't find the flies. If you go a little bit south, a little more temperate climate, you can find flies all year round, but they still see this large peak as you go into the field season where it peaks later in the season. Finally, on the West Coast in California and the Mediterranean climate, where we know it's very hot and dry in the summer, we often see a decline in population in the middle of the summer that's kind of spaced by higher peaks in the spring and the fall. All right, so what are the factors that drive this seasonality? Next slide, please. We've done a lot of work on temperature tolerances, both low temperatures in the winter and high temperatures in the summer. And on the bottom here, I just summarized some of the take-home messages. One, we found that spotted wing Drosophila goes into a diapause, which is a way for them to change morphologically and physiologically to help them survive cold temperatures. And they get a lot of that, especially in the northern parts of the United States. We also found that cool temperatures late in the fall, when they're developing that kind of a late term in fruit, lead to winter morph type flies, which have a characteristic morphology and physiology that again helps them survive the winter. And you can see the little diagram of the flies upper, the one on the right is a winter morph fly. They are more tolerating of low temperature, and the graph on the left shows how winter morph flies in orange and summer morph flies in blue respond to declining temperatures. And you can see even at -7 degrees Celsius, 50% of the winter morph flies are surviving. That's about, 20 degrees Fahrenheit. Whereas all the summer morph flies have died. But we do know that even winter morph flies are sensitive to cold and extended cold time periods. And the graph on the right is field data that we've collected over several years that shows that the more days below zero or freezing really reduces survivorship. And that kind of actually has led to some really interesting results where we see a correlation between how cold and severe the winters are, this is especially in northern parts of their distribution, and risk for the next season. And we think we can use that to help growers anticipate future problems. Now in the summertime, as I mentioned earlier, we know these flies really don't like really hot temperatures. And when they get above 90 degrees or so, they kind of go into a summer quiescence. that they will recover to them. That's kind of explains that depression in populations. Next slide, please. Let me move on to a really nice set of work that Devon Walton's group has been doing at Oregon that other people have also been helping him test. He's developed a population model and several models to explain the population growth of spotted wing Drosophila, and then has used that to test management option. And in this case, he's testing, what's the best timing for using your most efficacious versus your least efficacious materials. The first slide is model generated populations of the different life stages of spotted wing over the season when there's no intervention. The next one shows what happens if you go with your kind of least effective material early when the populations are low, and then save your best stuff for late. And you see some suppression, but it's not that great. And I would argue that a lot of us have kind of recommended this pattern in the past, save your best for last. Now, when Vaughn modeled the alternative option with doing with your best material first, next slide, actually showed in the model the best reduction in populations. They have now taken that model prediction and they can say, okay, let's test this under field. Next slide shows some results. Next one, sorry, Hannah. Shows some results, I think this is out of Oregon that Vaughn did, that does indicate that if you go with your best material first, the blue line, you get a little bit better reduction in infestation than when you go with your least effective material first in the orange line. Next slide, please. So these are just sort of the outputs of that model, and that bottom one is really the one we really want to continue to pursue because that may change how growers approach their rotation of materials. Next slide. Finally, I wanted to summarize some of the work that Joanna Chu at UC Davis has been leading from our team. She's looked at the genetic variation in different spotted wing Drosophila populations throughout the United States, and next slide, please, also in Europe and Asia, and asked how they compare. Next slide, please. She's done various types of analyses that are kind of shown and summarized in these principal component graphs on the top, but the basic take-home messages are, one, the spotted wing in the United States is sort of distinct from Europe and Asia. It's kind of interesting. In the United States, we see distinction between Eastern and Western United States and not a lot of exchange. But in the Eastern United States, there seems to be evidence of movement, longer distance movements of the genes and therefore flies from the southern part of the United States into the northern part. And this has real implications for how things like resistance might move around among populations, at least on the eastern part of the United States. Okay, I'm going to end it there, but I want to remind you, check out the full seminar to get webinar from last year to get sort of a more depth review of this kind of information.
All right, thank you, Greg. So I'm spearheading the next section in which we'll discuss host range and preference. And first, I want to highlight that spotted wing Drosophila host susceptibility is fairly well understood for crop hosts. I'm highlighting the bottom here from a study that was conducted by Dave Bellamy and others, indicating the relative preference of some of the main crop hosts initially that we were concerned about with spotted wing Drosophila, highlighting that through a series of different experiments, raspberry was the most preferred, followed by strawberries, blackberry, cherry, peach, blueberry, and finally grape. And this is consistent with what we've seen in laboratory studies, indicating that rosaceae or caneberries and strawberries are among the most preferred hosts. We know that when flies are given a choice, they prefer to lay eggs in raspberries over other hosts, and that in addition to preferring raspberries, they often seem to perform better in those hosts than in others. We also know that seasonality has a significant role in crop risk. And this is just an illustration that I use based on work that we've done trapping in North Carolina and then overlaying the crop harvest periods on top of some generalized trapping data. While we can catch flies during the entire year here in North Carolina, our populations remain pretty low until midsummer. And therefore crops that ripen and are harvested during these periods of the year, strawberries, for example, in spring, and some blueberry varieties that are harvested in the early part of summer, escape some of the larger population pressures that we experience later in the year when for us, caneberries are primarily harvested. Now this phenology is going to shift depending on where you are in the country and what crops you are growing. So blueberries may be a fall crop in other parts of the countries, such as the Pacific Northwest or the Upper Midwest. And in that case, they're going to fall into a higher risk period. So seasonality really influences the susceptibility of flies. But this fly trap capture pattern, where we see large peaks increasing very rapidly following midsummer, is conserved throughout the country. So we see relatively low populations, in some cases in cooler parts of the country, undetectable populations in the first half of the growing season, followed by a very rapid population growth in the second half of the year. We also know that fruit becomes susceptible to infestation when they first ripen, but the risk to underripe fruit can be decreased by decreasing populations. This is work that was done by a graduate student in my lab, Katie Swoboda-Badarai, where she looked at exposing fruit of different ripeness stages on the plant to ambient populations of flies. And what she observed was that when we had high populations in an unmanaged, unpicked research planting, infestation began as soon as that fruit started to change color, when it was green-pink. It was highest in perfectly ripe fruit, but that infestation potential occurred as soon as the fruit started to change color. However, when she repeated this work in a commercial field the following year, where the flies were actively managed through insecticide applications and fruit were harvested on a roughly 3-day schedule, there were low numbers of flies in the entire planting, and that resulted in infestation being concentrated in their preferred ripeness stage, ripe fruit, rather than these ripening fruit. And what we think is happening there is, under ideal situation, the flies would prefer to lay their eggs in ripe fruit. So when there are low numbers of flies, their infestation is concentrated there. As population density increases, however, we see spillover from that ripe fruit to less preferred unripe stages hierarchically. We also have learned that non-crop host range for spotted wing Drosophila is extremely broad. This was work done in Europe, and I really like this figure because it illustrates throughout the 12-month period of the year that spotted wing Drosophila was detected in at least one non-crop host species. So the take-home message here is that in most environments, there is potential substrate for these flies to develop on year-round, as long as the climatic conditions, the weather, are conducive to their activity. In addition, work done by Dara Stockton in Greg's lab up at Cornell has highlighted that flies will feed on many different substrates, not just fruit. And this includes things like mushrooms, flowers, and both mammal and bird manure. And so you can get eggs being laid and spotted wing Drosophila being reared out from all of these different substrates. They can also be found in compost piles or other decomposing substrates during the winter. And this is observations from Michigan during 2016 at 15 different locations, that pumice piles and compost were also a potential source of spotted wing Drosophila presence. But all these non-crop hosts are not necessarily created equal. And so while spotted wing Drosophila can readily attack lots of different possible substrates, their offspring, their larvae, do not do well across all of those different diets. And so fruit and mushrooms resulted in larger, healthier flies as compared to something like manure. But the take-home message here is there is something that these flies can eke their way through on most of the year when they are available, when the weather conditions allow them to become active. And so fruit is not the only substrate required for reproduction and survival. There's lots of other potential things that they can feed on as well. And this, of course, is going to change over time. And if we can understand where this host use pattern shift occurs, we can potentially target these flies when they are most vulnerable to management and their population sizes are the smallest. And so this is work that we're continuing in other aspects of this project. All right, now I'm going to turn it over to Phil Fanning, who's going to address pesticide efficacy and non-target effects.
Thank you very much, Hannah. So I'm going to talk about pesticide efficacy. So if you flick onto the next slide there. So when we think about pesticide efficacy, really we want to use our best products, like was pointed out with Vaughan's model, we really want to use good products early on, but generally we want to choose very efficacious products. So the slide you're seeing here is a kind of a breakdown or a survey that we've done of crop consultants, researchers, and industry folks across 9 different states and 19 different state crop combinations, where we asked them to rank different insecticides basically on their efficacy for spotted Drosophila based on their experiences with the crop. For the most part, we still recommend that, in high pressure situations, we make applications of insecticides at tight intervals, maybe every 7 days when pressure, SWD pressure is really high, make reapplications after rain, and choose effective insecticides. So if you look at this list, we're going to basically work from left to right here. You can see there's obviously some that we wouldn't recommend for spotted wing Drosophila as a sole application, and there's others that rank pretty high, above good and nearly excellent. So you can see the first ones, the neonicotinoids, they obviously ranked pretty low in this survey. Admire and Assail, which are 2 neonicotinoids, they ranked low. The next product, Cormoran, is actually a mix of Assail, which is acetaminophen, and Ryman, which is Nuvaloron. So this Cormoran product is a mix of both of them, and that ranked probably the highest of this first group that I'm highlighting here, which is neonicotinoids and premixes. I apologize for the international folks. These are all trade names associated with US products. The next group is the pyrethroids. These all have a PRY in the parentheses after the product name. And they all score good to excellent for spotted wing Drosophila control. So this has been an important chemical class for us in controlling spotted wing Drosophila. Spinosins, which is Delegate and Radiant, they also rank good to excellent in the listings here. The next, Lanate, which is a carbamate, is also an important insecticide. And then you have the organophosphates, all of which, with the exception of 7, also rank pretty high. Finally, we're going to finish with the diamides. There's a new product here, Verdiprin, that works really well for spotted wing Drosophila control. And we've seen some really good efficacy in our field trials. And then you have XRL, which has been on the market for a while. The last 2 don't really score as very well. Sphere, which is a biological product, has been tested quite a lot and does pretty well. So on the next slide, we're going to look at organic materials. And these organic materials are all OMRI listed. As you can see, we ran this survey with all of these products. So you're going to see that the levels are a lot lower here when it comes to the rankings. Entrust, which is probably our best product for spotted wing Drosophila control in organic settings, still only rates, around good. It doesn't meet that good to excellent ranking like a lot of the other insecticides. Obviously, Entrust does have some label restrictions that are really important when it comes to spotted wing Drosophila control. So there's seasonal limits. You're restricted to 5 to 6 applications per season, but there's also built-in requirements to rotate with other insecticides for resistance management. And obviously insecticide resistance with Entrust is something we're always very concerned about. So looking at these products, you have a lot of neem-based products. There's Azadirachtin and Azigard. Azira is a mixture of neem and pyrethrins. Then you have our Entrust, which is a really important one. And then some biologicals. Grandivo is a biological. Pyganic is pyrethrins. The other 2, Jetag and Oxidate, these are perioxycacetic acid products. I'm going to talk a little bit about them later. And then we have Veritran D. Veritran D, it's important to note, it is OMRI listed. We're still waiting for a label on a lot of fruiting crops, but we've tested that quite a lot. So on the next slide, we're going to look at this all in a different light. So what I've taken here is I've taken all of the rankings from the 6 chemical classes, the carbamates, diamides, neonicotinoids, organophosphates, and pyrethroids, and spinosins, and we're just highlighting the top one here. So obviously, when we're doing our insecticide applications, we're always advising to rotate our chemical classes for resistance management. So I'm not going to go through all this list, but you can see here if you were choosing a spray program based on the top ranked efficacy in terms of these products, you would choose the collection of these and rotate them for your insecticide class or for your insecticide program for the season. You can see here in the bottom, organic options. Again, you're kind of limited to entrust across all of these crops. All right, I'll move on quickly to the next slide, where we're going to talk a little bit about some of our work on feeding stimulants. So you can see here on the diagram on the right-hand side, you can see a little fly. So there's been some really interesting work done that shows that when flies like spotted-winged Drosophila land on a surface, basically they have sensors within their feet that can basically taste what they land on. And this is really interesting, and we thought it might be a really good way of getting spotted-winged Drosophila to uptake more insecticides. So we tested sweeteners, but we also tested yeasts, and yeasts play a really important role in spotted-winged Drosophila's development. When we rear it in the lab, we use yeasts in all of our diets. It impacts larval development and survival. We obviously use it as an attractant, and it also helps in adult fecundity. So yeasts are really important. So we tested yeasts and we tested sweetened surfaces in terms of sugar in the field. So on the next slide, we'll be able to look at some of our bioassay data from the lab. So just to walk you through the slides here, on all of these graphs that you see here, you will see proportion mortality on the y-axis on the left, and then across the bottom is the concentration of the active ingredient of the 4 insecticides we tested. So we had a neonicotinoid, acetamiprid, malathion, and then on the bottom we had 2 spinosins, delegate and entrust. And these were all products that we were very interested to see if we could improve their efficacy by adding in these feeding stimulants, sugar and yeast. If you go from the top right or top left across, you can see really acetamiprid benefited probably the most from sugar. In the dark line, you will see the active ingredient only. And then you have the fine dotted line, that is the active ingredient plus sugar. And then the other line, which is a dot, and a longer line is the active ingredient plus yeast. The real big take home here is the feeding stimulants did work pretty well in lab bioassays. Acetamiprid with yeast was probably the one we saw the greatest success in. Malathion as well showed some product. Interestingly, in the spinosins, we didn't see a big effect, but we did notice that when we added yeasts in with spinosins, it actually negatively impacted their efficacy. Looking at some field data in the next slide. This is some trials we done in northern highbush blueberry in Michigan. And the different color bars in this graph are different days after treatment. So they're different residue ages in the field. And on the bottom axis, we can see their products. We had spinetoram, which is delegate again. We had spinetoram and sugar, spinetoram and yeast, spinetoram, sugar and yeast. And then what we did is we'd spray these in the field, we'd bring them back into the lab and put them into a little bioassay container, which is the top picture on the left. And we'd introduce spotted-winged Drosophila and basically assess their mortality. One thing you'll notice is, for the most part, there was a lot of variation within our data, but looking across the treatments within the different colors, we can see that we really didn't see in the field a strong effect of these phagostimulants or feeding stimulants on at least spinetoram or delegates efficacy in the field. So we're testing some other products. As I said, acetamiprid looked really good in the lab, but it didn't really translate, or we didn't, at least in delegate, it didn't translate into the field. So the next products, or next slides we're going to look at is the crop sanitizers. So we were testing crop sanitizers because, as I said, we know that yeast is really important for spotted-winged Drosophila in terms of their development, they're attracted to it, and their survival. So in the field, we wanted to look at these crop sanitizers to see how they would impact control of spotted-winged Drosophila. So there's a number of different products. You have JetAg and Oxidate, Sporequill. I have Entrust on here mostly because we're looking at these a lot because we're looking for rotational partners for Entrust. In a lot of instances, these perioxic acetic products are OMRI approved, so they might have really good efficacy in terms of a rotation with Entrust. So we've done some trials in the field. I will say that the data I'm going to show next is purely associated with conventional products. And when we tested these, we did high water rates in the field, because we think coverage is really important when it comes to these crop sanitizers' efficacies. On the next slide, or on this slide, you'll see basically that this is a lab bioassay that was ran by Kelly Hamby in Maryland, where we looked at plated yeast within these Petri dishes, and we introduced in the perioxic acetic products into the Petri dishes. You can see here on the top left is the control with no impact. And then as we move to the right, we had a half dose of, in this case, it was JEDAG we were using, where we did see an effect on the yeast. You'll see that halo around that middle piece. And then as we moved up the doses, we saw we had a greater impact on the yeast. So in the field, if we move on to the next slide, you'll see basically how we tested this in a rotation. There's a few different treatments here, so I'm going to walk through it. The green is an untreated. This is where we treated no bushes. The next treatment was a rotation of Mustang Max, Lanate, and Imidan, so a pyrethroid, a carbamate, and an organophosphate. Then we had a pyrethroid followed by 2 applications of this JEDAG, or this perioxic acetic acid product. And then we had just a straight weekly treatment of JEDAG. So these were all done weekly as you see them here in the list. What you'll see for the first 3 dates, we didn't really see much difference between our treatments. And then our spotted wing Drosophila numbers really crept up. And you can see our untreated control in green on that August 14th date was really quite high. But when we look at the other treatments, we can see that our conventional rotation worked really well. But we also had really good efficacy from our pyrethroid and perioxic acetic acid treatment or spray program. And then our perioxic acetic alone product or rotation worked also pretty similar. So these were really some encouraging results. And we're really trying to delve into this more in some more trials coming up soon. I think now I'll pass you over to Rufus, who's going to talk a little bit.
Okay, thank you very much. So I've got a few slides to talk about natural enemies. Phil just talked about the direct effects on the pest, but as we develop our IPM programs, we really need to be thinking about side effects on these beneficial insects that are in our crop systems. So the data I'm going to share here are from a trial that we did a few years ago now, study we did a couple of years with colleagues in New Jersey. And we looked at this parasitoid, this predatory bug, and this lady beetle, and their susceptibility to different insecticides. You can see the methods there, and I won't go through the full details. You can look at the paper if you're interested, or just come back to this slide in the recording. Next, please. Thank you. So this shows data for fresh residues at the 0 day after treatment, and then those 14 days later. And the data on the left, you can see is percent mortality. So we've got the direct effect of the insects being dead, and then some that were knocked down, but they may have recovered, but they certainly weren't able to parasitize or attack any insects. So this is for the parasitic wasp, and you can see a range of different effects there, with most of the products that are used against spotted wing Drosophila being quite lethal to these wasps. Next. A slightly different effect on these pirate bugs. These were a little bit more tolerant of some of the insecticides. If you compare the 2 slides later in the recording, you can see that. But again, the broad spectrum insecticides are knocking out these beneficial insects. Next, please. And then the third group was the ladybugs. These may not have a direct effect on spotted wing Drosophila, but there's other beetles that might be predating on pupae of this pest down on the floor of a crop field. So I think it's relevant to look at these data. And you can see a few of the products that were quite toxic at the beginning may have lost that toxicity over time. So also interesting here to think about how the options used for spotted wing Drosophila might also be influencing this and the other natural enemies that we just showed. Next, please. We just heard about organic insecticides, and I think it's important that we recognize that the main tool for spotted wing Drosophila in organic production, which is in trust, is also quite toxic on natural enemies. This trial was done on a parasitic wasp that is not specific against spotted-winged Drosophila, but we would expect a similar result against the insects that we're hearing about, I think, in the next session of this webinar. So there are choices that organic growers can make on balancing this toxicity against the key pest versus the risk of knocking out these beneficial insects that we're so interested in getting into crop systems. Okay, next, please. And just to wrap this section up, we are starting to see in many of our crop systems around the country where spotted-winged Drosophila is being managed that there are some increases in secondary pests. Some photographs here show scale insects on crops. And I think this is something just to be highlighting to growers that might be on the call, that although you may be successfully managing spotted-winged Drosophila, we need to keep our eyes open and scout our fields for scale insects, mites, aphids, other kinds of insects that may be increased by the natural enemies being knocked out by these pesticides.
All right, thank you, Rufus and Phil. Now we're going to transition to discuss some of those biological control agents, and Kent Dana will be taking over for this section of the presentation.
Thank you, Hannah. So next, first slide. So I'm going to focus really on the classic biocontrol program that we're working on, although we are working with some of the native natural enemies as well. And this has been supported by the SCRI grant. But we're also doing this in coordination with a large international group, especially folks based in Europe and working with CABI. Next. So the first thing we do in a classic biocontrol project is to determine if there are natural enemies present here that give us support. We've got, going from left to right, this pupal parasitoid Pachygropoides, another pupal parasitoid Trichopria, and then different larval parasitoids. And what we found in most of the North American surveys prior to 2019, the pupal parasitoids are present, but they don't really give us control. Typically, it's below 10% parasitism. We are working with mass releases where we rear these in an insectary and release these in hoop houses. And we are getting a little bit of an impact of that, but nothing that looks like it's going to be economical. What's important to note is that we have very few larval parasitoids in North America. They're either not present, or if they do attack spotted wing, it's almost a rare occurrence. Next. This is because spotted wing has got this defense against the larval parasitoids that are present in North America. So they will attack spotted wing. They'll put their egg into spotted wing larva, but the egg is melanized. And you can see that in the upper left graph and the lower left graph. Those dark spots in the spotted wing are melanized eggs. So this is like a host defense. It encapsulates the egg and doesn't allow it to develop. So we get no parasitism. Next. So that leads to going to the origin of spotted wing, Asia, and looking for parasitoids that co-evolved with spotted wing that have overcome this host defense. So we've spent 2013 to 2017, mostly in China and South Korea, with a large group of folks helping us, looking for resident natural enemies, resident to the native area, that might help us control spotted wing Drosophila. Next. We found 19 species reared from Drosophilid species in both South Korea and China. Of these, there were 3 species outlined here in red that gave us greater than 5% parasitism. 1 braconid, Asobara japonica, and 2 fagitids, Ganaspis brasiliensis and Leptopolina japonica. Next. And here's a photo of those 3 insects. They don't have common names. We just go with Ganaspis, Leptopolina, and Asobara. We did learn from our collections in China and South Korea a little bit about their biology. We know that they attack the spotted wing early in the season, and they get outcompeted later in the season as the fruit is a little bit riper, a little bit more rotted. That's when you get more of the larval parasitoids that we find commonly in North America as well. Next. One of the other things I want to point out from our collections in China and South Korea, in the top graph, we see the composition of these three parasitoids. In red, that's Ganaspis. In blue, that's Leptopolina. You can see that Ganaspis and Leptopolina are the most common ones we're finding. You also see that at different collection sites, we find both of them together. That is good. That means they work in concert with each other. In the lower graph, we see the parasitism rate on 4 different host plant species, 2 rubus, a Fragaria, and a Sambucus. What I want to point out is that together, usually they were getting about 20% parasitism. We got as high as 70% parasitism at some sites. So my hope is we're not going to get complete control, but I'm hoping we get about 20% parasitism in North America. That might be enough to lower the population in some of these outlying areas, to lower the pressure of spotted wing going into the crop field, making insecticides more effective. Next. So how do we get these things from China and South Korea into the US? We have to do quarantine studies. We looked at a number of their biological parameters, temperature development, host stage preferences, and the like, interspecific competitions, but the real key is looking at their non-target impacts. Next. We see here Acebara, Leptopolina, and Ganaspis, and this is a group of different Drosophilid and other related fly species that we expose them to. We see Acebara was reared from most of these. These are offsprings per female. So Acebara is a generalist. It will never be permitted to be released in North America. Leptopolina did attack quite a number of species, but we can see it focused on this Drosophila melanogaster group, which includes Suzukii. And Ganaspis, and this is the G3 strain, really focused on Suzukii and closely related species. Next. So what's the process then to get this out? Well, the petition has to go through the USDA. It triggers a federal action, and that leads to 2 acts, the Endangered Species Act and the National Environmental Policy Act, or NEPA. Both of these address risk. Next. So in quarantine, we had to look at 7 different aspects of these parasitoids, biology and impact, and what would happen once we release these. But the most important is highlighted in red, and that's that non-target impact. Next. So we've submitted this petition to NAPO. Typically this takes 2 to 4 years. It's cleared NAPO. Next. And currently it is out for tribal consultation, and then we'll get into the public registration for public comment. Next. And that's when we ask you to make comments that this is important, spotted wing's a nasty pest, and that you do want these parasitoids to be released. The last steps, APHIS makes a decision, it goes through a FONSI Act. This process goes very quickly, so we hope to have permitting this spring and expect releases to start this late summer or August. And hopefully we get colonies built up and out to everybody. Thank you.
All right, so we're going to continue on the topic of non-chemical control tactics and turn it over to Ash Sayal, who's going to discuss cultural and post-harvest control strategies.
Thanks, Anna. Next, please. All right, so, the main goal of cultural and post-harvest control is to purposefully manipulate the environment in which pests live in order to reduce pest population and crop damage. Next, please. When we go out in the field and look at, for example, a blueberry bush, what we notice is that the temperature within the center of the blueberry canopy bush canopy is lower as compared to the lower or upper portions of the canopy. At the same time, the relative humidity is higher in the middle of the canopy as compared to the lower and or upper portions of the canopy. Now, when we look at SWD, oviposition in the field, that is based on our studies higher in the middle of the canopy as compared to lower or upper portions of the canopy. What these studies show that the temperature and relative humidity are the two most important factors that affect SWD survival and development in the field. For example, 68 to 86 degrees Fahrenheit is the optimum range for SWD. As soon as temperatures start to rise above that, the flies either don't survive or their reproduction gets affected. Similarly, relative humidity, they like to live in higher relative humidity. 70% or higher is better, but at lower relative humidity, they do not well. Now the question is, what can we do to manipulate these two factors to make the environment less favorable in the field? Next, please. And there are a number of things that we can do in the field to manipulate those two environmental factors to make the field less optimum, or environment in the field less favorable for SWD. Netting, irrigation, we can manage mulching in terms of weed mats, or pruning, harvest frequency, or using refrigeration of the fruit after harvest to actually manipulate these environmental conditions for SWD to make them less favorable and reduce survivorship and development of SWD. Next, please. Now, in the next few slides, I will show you a very brief overview of the work that was done under OREI project. For more detailed information on all of these aspects, please go, you can watch a webinar, the recording of which is available on YouTube. I will show you a link at the end of my slides. So let's start with the physical exclusion. Several studies were done in multiple states, and those studies show that if done right, mesh netting with using less than 1 millimeter mesh can exclude flies from the field. They can reduce fruit infestation and improve marketable yield, and also fruit quality in raspberries and blackberries. And specifically in blueberries, this exclusion technique can provide up to 100% control. However, because of the cost involved, it may not be feasible for large size farms, for example. And in general, costs run higher at the beginning. Some farmers have been very creative in repurposing their existing resources at the farm and using materials that they had at the farms to actually lower the cost to establish these tunnels at the farm. Next, please. So irrigation is another factor that we can manipulate to actually affect SWD populations in the field. For example, there are two main types of irrigation. One is drip irrigation, and other is overhead sprinkler irrigation system. And we compared both systems. The relative humidity was lower in the field when drip irrigation was used. As I pointed out earlier, SWD is very sensitive to low humidity. So this comparison of the two irrigation systems did is showed that the SWD fly emergence was lower in the field where drip irrigation system was used as compared to the fields where overhead sprinkler irrigation system was used. Next, please. Mulching is another factor. For example, here, when we look at biology of the flies, when larvae feed obviously within the fruit, when they're fully developed, they drop out of the fruit onto the ground and actually pupate inside the soil. When we use this mulches on the ground floor, actually what it does is it prevents the larvae to get inside the soil to pupate, and overall surface temperature on those mulches is relatively higher, which results in majority of the larvae being killed due to heat. And also as a result, they cannot compete with development and fruit infestation reduces. There are many types of mulches that are available. Black weed mat is most common one, which was very effective. And another reflective mulch was that was tested in Michigan, for example, this Mylar, that also showed significant results against SWD. Here is a publication. If you are looking into or interested in more details, please take a look at this publication from our group that has details of studies done in multiple states. Next, please. Pruning is another important factor for SWD. What it does is basically heavy pruning opens up the canopy, lets more light pass through the canopy, which slightly reduces relative humidity and increases temperature, which creates less favorable environment within the canopy where most of the fruit is. As a result, overall fruit infestation decreases. Studies in multiple states have shown that this may be an effective way to actually lower population of SWD flies in some situations. In addition to direct impact on SWD, what it does is actually improves spray coverage as well as harvest efficiency, which also is good for overall production at the farm. Again, the details of these results were published in this publication shown here. If you are interested, please, in details, please take a look at this publication and get more information there. Next, please. Harvest frequency. You know, harvesting is very important, especially when we're dealing with pests like SWD, which is attracted to the ripe fruit, as Hannah showed in an earlier slide. When we leave more ripe fruit, it serves as a resource and attracts flies more toward the field. When we remove this resource, it makes the environment within the field relatively less attractive. As a result, overall infestation reduces. As studies show that harvesting every 2 days was more economical in terms of marketable yield, and also the overall fruit infestation and in terms of cost of actually per unit fruit harvested. Next, please. Another most important factor is, which is present in almost every field, is the cull fruit or leftover fruit in the field, which obviously is really overripe and very attractive to flies and serves as a breeding ground for those flies. If we can, several studies show, if we can remove and destroy those cull fruit, there are different ways to do that. And for example, you can put them in the sealed plastic bags and leave them in the sun up to 2 to 3 days. That will take care of that. Or you can bury them at least 2 feet deep into the soil. That will also take care of that and will prevent further fly emergence from those fruit. This is extremely important. This will reduce attraction of the flies toward the field and overall lower infestation rates in those fields. Next, please. You know, last but not least, if you have harvested a fruit and you suspected there may be some fruit infestation there, you can still do something, and that is refrigerating fruit at lower temperatures between 32 to 36 Fahrenheit. If you do that, that will do a couple of things. Number one, it will kill most of the larvae inside the fruit. And number two, it will slow the development of the fruit, which will allow you more time to bring this fruit from field to the table, through the marketing channels. So if there's a way to maintain lower temperature throughout the supply chain, that will significantly help lower the larval development inside the fruit and bringing clean fruit to the table. So to summarize, you know, there's a number of cultural practices that all may be applicable in every situation, but a good combination of these strategies is an excellent way to supplement your other chemical control to overall decrease population of this fly in the field. this was just a quick overview of what we have done under OREI project on these cultural controls. For detailed information, here is a link to our webinar on organic management of spotted wing Drosophila, which is available on YouTube. At this link, you can access that and get that detailed information on all these aspects. Thank you.
All right, next we'll be moving on to an update on monitoring research and potential applications of that information for behavioral control with Cesar Rodriguez-Zona. Cesar?
Thank you, Hannah. So our priority in SWD monitoring has been to identify a lure or bait that was effective, selective, that provides early warning, and that correlates with a fruit infestation. In a multi-state, multi-crop effort to achieve these goals, we tested various lures that combine fermentation lure used as our standard lure with volatiles from yeast and leaves. However, we found that combining volatiles from different sources made the standard fermentation lure less attractive to SWD. Next. We also monitored SWD attraction to these lures during and after the fruiting season and found that the yeast and leaf lures are more effective when leaves and fruit are absent, indicating that background odors may affect SWD response to these lures. Next. To date, the lures based on fermentation odors remain the standard for monitoring SWD and are commercially available in the US. Although these lures are effective and provide early warning in some states, they are not selective and not always correlate with fruit infestation. And next. To make traps easier to use, we have tested the efficacy of red panels to capture SWD instead of using the standard liquid traps. In a study in Michigan in cherries, red panel traps were better at capturing SWD than the liquid traps. In New Jersey blueberries, red traps and liquid traps capture the first SWD before fruit infestation, thus providing similar early warning. However, studies conducted in various states found variable results. And next. A mark release recapture study was done to determine the range of attraction of these red panels. This study found that the maximum dispersive distance for SWD is 90 meters, which translates to the need of 1 panel trap every 7 acres. Because there is no trap lure and lure that fits all geographical regions, stakeholders expressed the need to move towards implementing behavior-based control methods. We have tested a splat attract and kill formulation from ISCA Technologies that combines attractants with insecticides in a waxy dollop that releases the attractants slowly. This formulation shows promise in reducing SWD infestation. In addition, we are testing a food-grade gum matrix that reduces SWD oviposition in fruits by competing with fruit for oviposition sites. These two formulations, the splat and the food-grade gum, are going through the process of registration. Moving forward, we plan to continue to compare the efficacy of these behavior-based strategies in multiple crops and states. We also plan to combine them with repellents to develop novel push-pull systems. And we also plan to determine the economic feasibility of these strategies to increase their adoption. Thank you.
All right, and our final topic puts all of these pieces together in order to make efficient decisions. So I'll turn it over to Miguel to tackle that topic.
Thank you, Hannah. So our team focuses on understanding the economics of SWD and how to better control. As part of this, our team has evaluated different control strategies, in particular comparing calendar-based spraying, which is the status quo, what most growers use, versus different IPM strategies, including early harvest, including monitoring to guide insecticide applications and including post-harvest cooling. So one thing that we have learned in the past 5 years is that the economic assessment is very specific to the region and to the crop and to the particular problem with SWD. So what we have been focusing on is also to developing Excel-based interactive management support tools to develop specifically for regions and crops to support decisions on how to best control. Next, please. So I will share with you some examples of studies that we have done. The papers associated with these studies are available in our website. And for example, in New York, we focus on high bush blueberry farms, focusing mostly on UP operations and sell direct to consumers. And in this case, we compare calendar-based sprays strategies to monitoring-based IPM strategies. The main finding here is that in order for monitoring-based IPM strategies to work better, it's very important to increase the efficiency of trapping of traps. Because current trapping efficiency works in favor of, is so low that it works in favor of farmers applying insecticide through the cropping season. So our results here is that one way to make monitoring more economical is to improve efficiency. We find, however, that some of the monitor-based strategies have slightly lower total costs than calendar-based spray strategy in the case of blueberries in New York. However, important is to consider that when we consider the environmental costs of insecticide application in terms of water pollution, effects on birds populations, and even effects on resistance of SWD to insecticide application, we find that most monitoring-based IPM strategies exhibit lower total costs when we take into account these costs. This begs the questions of the need for incentivizing farmers and growers to use IPM strategies. Next, please. So another example is we focus also on high bush blueberries in North Carolina. And in this case, we focus in the comparison between not having and having post-harvest cooling. And one of the benefits of having post-harvest cooling is that you can avoid insecticide application and solve the problem of infestation, of presence of larvae in the fruit after you harvest. These models are complex because problem is as you keep fruit on storage, especially when you sell to the fresh market, the price of the fruit declines over time. So basically our results indicate that it may be a good option for growers in particularly in North Carolina and perhaps in other areas to consider post-harvest cooling as a strategy to control for SWD. We find, for example, in this case that the break-even year of investing in a cooling facility is in year 4. After year 4, you recover your investment and it becomes profitable to have cooling. We also find that once you have the cooling equipment, if you use post-harvest cooling as a strategy to control, then the average profit increases by over $100 per acre, about $50 per over $100 per hectare, which is about $50 per acre. Next, please. Another example is in low bush blueberries in Maine. And here we focus on comparing control strategies during weeks that are close to harvest. And in particular, we are evaluating whether it is worth to spray one more time or to harvest early. And our results are very interesting, even for those growers that are not organic, that use insecticides. It may be harvesting early, leaving a small portion of the fruit unharvested may be economically beneficial in comparison to harvest 100% of the fruit and apply insecticide every week. For those growers that are constrained because they cannot apply insecticides, we find a range of early harvest that ranges between harvesting when 65% of the fruit is ripe to almost 99, depending on the impact of SWD on yields, the price, and other considerations. Next one, please. And this is the last one. So given that the problem of controlling SWD is very specific to crops and regions, our strategy has been to develop interactive management support tools. They are Excel-based, easily downloadable by users. I am showing you, for example, the one that we developed for low bush blueberry production for Maine. The yellow screen basically tells the grower, give me the characteristics of your farm, of your production facility, and the price that you will get. And we have a few crop budgets, and then the purple or blue screen on the right shows you what will be the return to either early harvest or apply insecticide in a given week under different scenarios. Next. So that concludes. I just want to mention that in the future, we are focusing on developing more Excel-based interactive management support tools for different regions as we learn more from the scientists about the impact of SWD. And in particular, we are focusing this year on the following comparison. We are focusing on comparing monitoring methods, trapping versus fruit sampling, which is becoming increasingly popular. We are going to examine the economics of biological control. When is it profitable, under which conditions? And for some growing regions, we are also going to assess the economic benefits of netting. And that concludes. Thank you for your attention.
All right, so that concludes our webinar for today. Just a few take-home messages for each of the sections. I'm not going to belabor the point by reading through these, but these are useful synopses of all of the different topics that we covered. I want to highlight some additional online resources. So there are websites that have been produced by members of our project team, as well as our project website, swdmanagement.org, where the recording of this webinar will be available by the end of this week. We also have social media tools for our project that you're welcome to engage with us on, as well as some state-specific resources that are highlighted. We're going to spend some time now answering the questions that came across in the chat box as well as in the Q&A. For those of you who can't join us or who have to run off to another meeting, feel free to email your questions to me at this email address, and I will make sure the right member of our project teams gets directed those. So now I'm going to go ahead and stop sharing my screen so that you can see our panelists and I can direct some of your questions to them. All right, so I'm going to go ahead and answer a few of the questions that came in through the chat box. Before I do that, I want to point out that several of the speakers from this presentation are also presenting here at the NARBA meeting. So later this afternoon, Kent Dana will be talking more about biocontrol and SWD in greater detail. And tomorrow, Von Walton will be talking about some non-chemical management some novel management tactics that his group has been focusing on developing for spotted wing Drosophila. In addition to those folks, Jana Lee, who's with USD ARS at Oregon State University, is going to be presenting on some work that she's been leading in collaboration with researchers developing sterile insect strategies for managing spotted wing Drosophila. All right, so I'm going to go through the questions that came in the chat box during the presentation. We have about 5 minutes remaining in this session to answer questions, but while I'm going through those, please also feel free to type additional questions into the chat box. So first question that came in was asking about if I could comment on the potential for cultivar resistance to spotted wing Drosophila. And we know from work that was done by a range of different groups that flies generally prefer to lay eggs in softer substrates than in firmer substrates, meaning that they like softer fruit rather than firmer fruit. They also work that has been done, and this is the work that was done by Kent Dana out in California, has demonstrated that in peaches, flies don't want to lay eggs in peaches with the fuzz on them. But if you shave that peach, you can actually get more fly egg laying to occur. And so there are these physical characteristics, firmness, and then fuzz or pubescence on the surface of fruits that can deter fly egg laying. So in the case of peaches, flies will go ahead and reject peaches. In the case of firm fruit, flies will often attempt to lay eggs and eventually lay some but fewer eggs in those fruit. That being said, I am not aware of any breeding programs that are working specifically on incorporating traits for spotted wing Drosophila into their breeding strategy, because those are tricky things that come with a lot of other constraints associated with them. And it's a bit unclear that just by reducing preference that you might decrease actual infestation in the field. So these are an interesting biology component that's useful to understand and might have some impact in the future in reducing the likelihood of infestation. But at this point, there aren't the really clean, obvious targets to reduce infestation through breeding. The next question that came in was asking about cold storage, post-harvest cold storage, and the impacts on fly survival. And so that's work that was done in my lab. And the question specifically was asking how many days at 32 to 36 degrees Fahrenheit should berries be maintained to kill spotted wing. And there's a little bit of variability depending on the life stage that you're talking about, whether you're talking about eggs, small larvae, or large larvae. But kind of across the board, we tend to see infestation decreasing at 34 degrees Fahrenheit or cooler for 3 days or longer. If you go for shorter than 3 days, those eggs and larvae will stop developing when they're at their cool temperature. But when they come back up to a temperature that they can continue to develop at, which is, for us, room temperature, 68 degrees Fahrenheit, they will continue to develop. They won't necessarily be dead. So 3 days is when we start seeing an increase in mortality after being held at temperatures of 34 degrees Fahrenheit or cooler. We see higher mortality the longer you hold fruit at those cold temperatures for. So we've held them at both 34 degrees Fahrenheit and 32 degrees Fahrenheit for 3, 4, and 5 days. And you see greater mortality when you go up to 5 days. That's as long as we've gone, because we wanted to select temperatures and durations that were potentially feasible for growers to implement. But that also has obviously implications for supply chain as well. So if you can keep it at that cold temperature throughout the supply chain, that's going to further reduce survival. The last question that I wanted to tackle that came in prior to this session was whether or not there was government inspection or care for imports associated with spotted wing. And spotted wing Drosophila isn't a quarantine pest in the US or in Europe. And so for people who are growing and marketing fruit in those areas, it's not something that your government is going to be targeting sampling for. So really the responsibility for tracking damage due to spotted wing Drosophila is mostly at the grower and the marketer level. And that's really where we've been focusing our efforts on developing tools that can be utilized by those groups to ensure that fruit remains undamaged. And then the last question that's in the chat or in the Q&A is how many acres or how many growers went out of production because of spotted wing Drosophila in the United States? And I cannot give you a good number on that. It's a really good question. One of the tricky things about getting at that is the growers who have stopped growing are no longer going to be coming to meetings like this and responding to surveys. So a lot of what we've heard for impacts on farm viability due to spotted wing has been from anecdotal evidence. So stories in trade publications, stories in local newspapers, growers telling us that they have reduced acreage of a given crop or removed a given crop from their farm. And so I have personally spoken with growers who've reduced their acreage of later fruiting or later harvesting blackberries, primocane varieties in North Carolina. They're shifting their primocane production to early rather than to floricane production rather than growing them as primocane varieties. And then also stories from raspberry growers in the upper Midwest who've reduced acreage or elected not to be producing raspberries as much. So if you guys have information on that, I would be very interested in you sharing that. So if you have reduced acreage or you know of friends, neighbors, colleagues who've reduced acreage or gone out of production for certain crops, that would be really good for us to know because we'd like to reach out to those folks and figure out if there's ways that we can be of assistance. All right. Those are all the questions that I see in the chat box, and we are right at time. So I think we can go ahead and wrap it up. Please do reach out to me either in the conference app or at my email if you have additional questions. All right. Thank you all.